Hi everybody, so welcome to this video regarding RNA extraction and RNA integrity analysis using gel electrophoresis. Just so you know that RNA extraction, we use a chemical known as triazole, very hazardous, so make sure you read the risk assessments and cost forms and sign those and read those before you get started, of course, with all the reagents that we use. So RNA extraction requires key steps where we extract the RNA from our cells during high versus low calcium-induced differentiation. This method allows us to extract RNA, but in the future, if you ever wanted to extract RNA, DNA, and proteins, this method allows you to do it. It's not the best method for all of them, but it allows you to do that. So in this case, we're just using triazole into our samples, and this one allows us to extract RNA. And the process is very simple. Initial video will be showing you just adding or precipitation of our RNA. So we use a chemical known as BCP or bromochlorophenol, which basically allows to separate DNA, RNA, protein, and any kind of organic material that might be in there because it's coming from cells. The second step is to very carefully actually just to take the upper aqueous phase, which will contain your RNA, which is a clear phase, and then what you do is you precipitate it by adding isopropanol, and we also use glycoblue, which binds to DNA, RNA molecules, and you see a blue dot at the end, which allows you to distinguish where your RNA is without losing it. Remember, RNA DNA is opaque, so you want to know where it is exactly, and that allows you to do it. And then what you do is you wash it multiple times and resuspend it in water. I'm just going to mention those 3 steps specifically in the next few minutes, and of course the protocol itself has additional steps such as centrifugation, washing with ethanol, for example. I will go through with you at a later date or on the day. And just remember, these are the key steps I'm showing you today. Along with this, I'm going to show you a short video how to make gel electrophoresis. So that's a really nice video because most of you never made a gel before, because it's usually made before you get there into your labs. And what it should look like when you run it, what the RNA should look like once you've extracted it and analyzed it in gel electrophoresis. So hopefully this is helpful, and also you're welcome to email me any questions. So let's get started. As you can see, Max has now already added the BCP, bromochlorophenol, and you can see it's very cloudy as you centrifugate it very harshly for about 15 seconds on the vortex, and it should become cloudy and become one.
So I'll tell you before you video, but you can see, as I was saying, the pink is the protein layer, the cloudy white layer is the DNA, and the clear colourless layer is our RNA. Okay. So this bit's quite tricky. So I want to just take the clear colourless layer without disturbing the DNA layer into a new tube. Much of it as you can without disturbing that.
And the final step is basically adding the isopropanol and the DNA. You can see it right there.
To make an agarose gel, first you need to set up your apparatus. Start by taping the ends of the gel casting tray with autoclave tape. Make sure there is a good seal between the tape and the edges of the tray to avoid leaks. Lay the gel casting tray on the bench and insert the gel comb. There are 2 different types of combs that will make different size wells to run your samples. Make sure you choose the size that fits your needs. You should place your comb at the top end of the casting tray, or if you would like 2 sets of wells on the gel, you can place a second comb in the middle of the tray. Next, you need to prepare your agarose. First, measure out the required volume of 1 times TAE buffer. For this small casting tray, you need 50 ml. Pour this into a 250 ml flask. Now you need to weigh out your agarose powder. This should be done in the weigh safe to prevent inhalation of the powder. Turn the weigh safe on at the side of the box, then turn on the balance and wait for it to calibrate. Once calibrated, place a weigh boat on top of the pan and zero the balance. Weigh out the appropriate amount of agarose. For most experiments you will want to make a 1% agarose gel, therefore here you will weigh out 0.5 grams of agarose to add to your 50 ml of buffer. Once you have weighed out the agarose, remove it from the balance and clean your spatula and the balance with Blue Roll and BioCleanse solution. Make sure you turn off all of the equipment as soon as you have finished with it. Now carefully pour the agarose powder into the flask with the buffer and gently swirl to mix. To melt the agarose you will use a microwave. Set the microwave to run for 1 minute, but check the flask after 20 seconds and then more frequently as the agarose heats up and begins to melt. Never leave the agarose unattended in the microwave and do not allow this solution to boil vigorously. Make sure you have a heat resistant glove as the flask will get hot. To check whether the agarose is melted, remove the flask from the microwave and gently swirl it to look for particles in the buffer. When the agarose has melted sufficiently, you will see a clear solution with just a few grains floating in it. Here you can see the solution is still cloudy and therefore not fully melted. Here you can see the solution is now clear and fully melted. Carefully take the melted agarose back to your bench. Now you need to add CyberSafe so that you are able to visualize your DNA after running your gel. CyberSafe is supplied as a 10,000 times solution, so in this case we would add 5 microliters of CyberSafe to the agarose solution. Once the CyberSafe is added, gently swirl the flask to evenly distribute it within the melted gel, then carefully pour the gel into the casting stand and check for any leaks. If there are no leaks, then leave the agarose on the bench to set. You will know when the gel is set because it will become cloudy. Once the gel is set, carefully remove the autoclave tape and place the casting tray inside the gel tank. Carefully and slowly remove the comb, making sure that you lift the comb straight up to avoid damaging the wells. Now add a few hundred mils of 1x TAE buffer to a jug and carefully pour this into the gel tank. Fill the 2 reservoirs at either end of the tank and then finally add a small amount of buffer over the gel to the fill line on the side of the tank. Next, add your samples to the wells in the agarose gel. Carefully pipette the samples by hovering your tip at the top of the well and gently depressing the pipette. Try not to touch the agarose gel with your pipette tip as you may break it. If you have shaky hands, it is often helpful to use your other hand to steady your pipetting hand, as shown here. Once you have loaded your samples onto the gel, you should fit the lid firmly on top of the tank, making sure that you are connecting the red side of the lid to the red side of the base. Then insert the cables into the power pack, again connecting the red connector to the red socket and the black connector to the black socket. Set the voltage to the required number, often this is 100 volts, and press run on the power pack. You should see bubbles emerge from the wire on the black side of the tank. Your DNA will run from the black side of the tank to the red side of the tank, so always check that your samples are loaded near the black end of the gel tank.
Just to show you what the gel would look like as it would run, this is a still image, but just to show you that the loading die that you include in your gel will allow you to visualize over time the migration of your samples. So this should give you an idea of how far the samples have run, and you want to just make sure that they run approximately 3/4 of the way of the gel to give you better resolution. And again, this goes back to the product size we would expect, and again this links to the worksheet that I've included as well, so have a look at that at the end, and just make sure that you're aware that this separation is important, and the longer the better over time. We visualize the gel using the gel dock. If you remember what I mentioned earlier in the video, that as you come into the lab, the gel's already made, but they contain a very specialized chemical or a component known as CyberSafe. CyberSafe allows which basically binds to double-stranded DNA, so this is your PCR product, and it intercalates and binds, and when it's excited by UV, which is gel dock system will do now, and we'll demonstrate it later with an image, is that when it's excited by UV, it actually expresses, or you can detect it under this gel dock system. So again, just to give you an overview, so CyberSafe is added to the gel, which binds to double-stranded DNA, and when it's excited through UV, it leads to the detection and visualization of the bands, and then we can do the analysis of our bands within that. So the question to ask yourself is, once I've extracted RNA, how do I know that I have intact RNA? So intact RNA means it has good quality, usable RNA, which you can use to amplify complementary DNA, and that's the next step, of course, which I'll go through later. And that template is used for qPCR. So if it's degraded RNA, then why is that a problem, first of all? And the consequences of degraded RNA means that we can't use it. So what's the difference? So the visualization is quite obvious. So we've got intact RNA here. In the third lane here, you can see two of the strong ribosomal S subunits of 28 and 18S. These show that the RNA is intact, is good integrity, and is usable for your cDNA synthesis and qPCR analysis. As you can see with the degraded, it's got a stark difference between the two, and there's no substantial banding at all really between the 28 and 18S. And the degradation is really the degradation of a sample is really kind of effective on your results, because all the RNA that you've got is actually no longer a valid sample to amplify from. So basically what I'm trying to say is that the degraded RNA could give you false data if you decided to go to do cDNA and qPCR analysis. The data is not reliable. So what you're looking for is these two specific bands. Also you get these small microRNA bands down here, which are about less than 100 base pairs, and if you remember from your reading that the precursor microRNAs can range from 70 to 100 nucleotides long. So this is what the microRNAs, or your microRNA of interest, that we could be looking at potentially are down here. So in reality, I'm hoping that you'll get this sample here for all your samples, but if you get this, that means we have to throw away or disregard it for analysis. So thank you for listening. Hopefully this video was helpful. Any questions, as always, you're more than welcome to email me. Thank you.